It's my pleasure to introduce Dr. Joni Rutter. Joni is the director of the National Center for Advancing Translational Sciences, NCATS, at the National Institute of Health. She leads a diverse portfolio of programs designed to overcome scientific and operational barriers that slow the development and delivery of new treatments and health solutions. Under her leadership, NCATS advances innovative tools, platforms, and strategies that make the translational process more efficient and effective, accelerating research across a broad spectrum of diseases with a particular emphasis on rare diseases. By strengthening the science of translation, NCATS helps transform promising discoveries into real-world applications that improve human health. Previously, as NCATS Deputy Director, Dr. Rutter built and strengthened partnerships across the government, academia, industry, and nonprofit patient organizations, helping to align stakeholders around shared translational goals. She is nationally and internationally recognized for her contributions to translational science and rare disease research, and is the recipient of multiple honors, including the 2022 Rare Voice Award for Federal Advocacy, the 2022 Fed Health IT Women in Leadership Impact Award, and the 2025 Charles A. Sanders MD Partnership Award from the Foundation for the NIH. Dr. Rutter earned her PhD in pharmacology and toxicology from Dartmouth Medical School and completed a fellowship in the Division of Cancer Epidemiology and Genetics at the National Cancer Institute. It's with my great pleasure to introduce Dr. Joni Rutter.
All right. First of all, I want to thank SEMA for really starting off this day so well and setting the stage for this conversation. And really putting a spotlight on how we should be thinking about measuring translational science and translation as a discipline. And also providing this great venue for having this discussion today. I also want to thank Jeff and Patrick and all of the meeting organizers for the opportunity to join you today. I asked them what they'd like to hear about from me as part of being part of the public sector here. And so what they said to me was, hey, look, we'd love to hear about new ideas on scientific funding and publishing and enhancing reproducibility and rigor in research and helping funders create and spend dollars more effectively. I thought those sounded like terrific ideas. So I'm gonna touch on all of them. These topics are timely, they're relevant now more than ever before, because really we're struggling. We're struggling with this. And to keep a competitive advantage, as Seema talked about so eloquently, to keep that a competitive advantage at the global scale, this is a really important conversation. And it can't end here. It has to keep continuing, and we need to do something about it. So as Seema kind of alluded to, I'm going to reiterate what she said, is it's not that we really have a discovery problem. We do a really good job at that. And I think the last panel, they said the same thing. It's essentially that we have an architecture problem. The science is moving so quickly that we're not able to keep up with it. And translation in and of itself remains slow, it remains fragmented and somewhat bespoke even. So I'm going to touch on all of those things that they asked me to touch upon, but I also first want to tell you a little bit about who I am and the organization that I come from. I run an organization called the National Center for Advancing Translational Sciences, also known as NCATS. It's much easier to say. And it's at the National Institutes of Health. We are a part of 27 different institutes and centers at the National Institutes of Health. And NCATS exists to help address exactly the types of problems that we're talking about today. But unlike most of the other institutes at the NIH, NCATS doesn't have a specific disease or an organ system that we really focus on. We study all of them. And the idea is that we want to understand how to bring more treatments for all people more quickly. And we innovate through infrastructure, which may not sound intuitive, but that's how we think about things. So I want to just start off by hopefully giving you a little bit more about sort of our public health mission. The public health mission is essentially this that we're trying to address. The daunting task for us is that there are over 10,000 diseases. Rare diseases make up most of them. Only about 5% of those diseases have treatments or cures. That leaves 95% of opportunity. So that's the challenge. And it's a challenge because this number has been this way for decades. So the challenge is, can we change this number in the next decade? So I think we can. But part of the reason that we have this problem is because the time and the cost that it takes to do research, it can take over a decade to move from a promising sort of therapeutic candidate, for example, in the preclinical stages, to bring something to the medicine cabinet. Over a decade, $2.6 billion per year spent doing that. So that's a long time, a lot of money. But the big killer of this activity is that 9 out of 10 times, those promising therapies that enter clinical trials fail in phase 1 or phase 2, largely in phase 1 and phase 2. They fail in phase 3 too, but not as much. So there are lots of reasons for that failure. And part of what NCATS does is to work to understand those failures. And oftentimes they're really not failures per se, but they are sort of bottlenecks and that are very difficult to surmount. So we kind of give up, or we don't think about challenging them and changing them. And I think the time is now that we start challenging them and changing them. So our goal at NCATS is to be the bottleneck breakers, if you will. So in many ways, our mission is shown here, and you'll see some keywords that have been sort of make it the stars of the show today, but essentially developing new approaches, demonstrating their usefulness, and then disseminating those findings. So the 3 Ds that we've been talking about today, we have been thinking about this as well. And our mission is really to turn research observations into health solutions through translational science. And our idea is that if you solve one project, that matters. That does matter. But if you solve a recurring bottleneck, you change the field, and perhaps you change the many lives that are affected by what you're doing. So these are the kinds of things that we do at NCATS. Essentially, the idea here is that we want to re-engineer the translational pipeline. And so what we show here is if you think about this as a pipeline, the translational science pipeline, from preclinical to clinical activities, there are many things that block that pipeline. They can be administrative, they can be operational, they can be scientific, but most often the real problems are those operational and scientific problems, operational and administrative problems. The scientific problems are usually surmountable. We need the right technologies, the right measurements, et cetera, but those are often surmountable. Operationally, though, those are the biggest challenges that we see in translating these findings to the next stages. And so we want to develop the tools and the technologies and the partnerships that really help us address those longstanding bottlenecks that make it difficult to go to the next level of the translational process. So the big issue is that science is accelerating, but the operating system that funds, that validates, that publishes, and translates it remains somewhat sequential. It remains somewhat bespoke, and it remains somewhat fragmented. And so that's part of the issue around the bottlenecks that we're trying to solve here. So the list that I show here, these common bottlenecks that I show on the slides, I realize I'm pointing right here, but they're all around us. This is a list of bottlenecks that we've been working on to help solve. But what we're really striving to build for is a shared infrastructure that can be compounded, that we can create as a force multiplier. That shared infrastructure as a force multiplier is exactly what we're meaning to do, so that people can leverage and build from it. We want to generate knowledge that accumulates, not knowledge that's fragmented. And we want translational systems that run in parallel. We don't want them to run sequentially, because when they run in sequence, then if something fails before it, then it's going to slow down after it. So we want them to run in parallel. So these are some of the ideas that we're trying to address through NCATS. So I want to talk a little bit about how the public sector is a big part of what I'm talking about in this keyword that was just relayed in the last panel, this keyword of an ecosystem. The public sector has a big role to play, but it's very different. The public sector excels at developing, the private sector excels at developing products. The public sector, it has a different role. We build the shared infrastructure that no single company can do or would want to do or could even justify doing alone, but that every company, every academic organization, every sector could actually then leverage. And so that's the importance of the public sector in this framework of the ecosystem. And as an ecosystem, we need to think about how we fund shared capabilities. And so at NCATS, we think about the preclinical and clinical translational pipeline as part of this sort of general capabilities that we want to support. And so we have general capabilities around drug discovery, preclinical development, and large programs that support that key infrastructure. And on the clinical side, Seema mentioned the Clinical and Translational Science Award Program, or the CTSA program. This is a large network of academic medical centers across the country. It's the largest program that we have at the NIH, and it's there to build the infrastructure to support clinical research and clinical trials. And a lot of that work doesn't just come from NIH-funded studies. That infrastructure is built and leveraged by biotech and industry as well. So this is an important factor for the clinical arm, is developing these clinical networks to be able to leverage what's needed for the community. And if we can think about this as an ecosystem, we need to think about how we fund shared capabilities, not only just individual projects. And we need to better reward sort of this how we reuse outputs, how we set standards, how we hold people accountable in using those standards, how we build platforms, and how we publish all data. All of those factors are key in making sure that we can really advance the science that we need to advance and stay competitive in this very competitive environment. The key thing to consider is how we diversify our biomedical portfolio. And we need to match that capital to the problem. So public and philanthropic funding is absolutely necessary for taking on sort of these high risk, high cost kinds of activities for those public goods. And then that private capital can then be better invested and as it aligns and it matures with the products that they can scale and find opportunities for those products to scale. So if we invest well, our funding and our innovations, they go farther, faster. And so I want us to think about not just translation as an ecosystem, not just as a pipeline, but really as a multiplayer enterprise, and where and how we direct those resources, where we and how we direct the capabilities, how we do training of the next generation to pick up the ball when we need that to happen. These are all factors in thinking about this as a multiplayer game. So in terms of pre-competitive infrastructures, things like measurements, things like standards, things like data, platforms, clinical networks, regulatory science, trial readiness, all of these things should be things that the public can help support and fund so that they are available and can be leveraged by the private sector and other ways of building that capital. So I want to also just turn a little bit to in sort of rethinking even how we do this in publishing the literature that we have and publishing the findings that we have. At NCATS, we think a lot about publishing, and one of the key things that we find is that the literature is really good at finding positive results, but it's not very good at finding negative results, and that's a problem. So what we did to address this problem is we coined a new program that we call LitCoin, because literature is the coin of the realm. So some people don't like this name, but we're stuck with it for the moment. Happy to change it. But the whole idea is that we want to create a machine-readable, sort of AI-ready ability to sort through literature. Both retrospectively and prospectively. Now, doing it retrospectively is a little bit more difficult, but prospectively, we can at least start there. And so what I'm talking about here is the LitCoin process involves a researcher taking an observation at a discovery that they've made during the course of their work, and they write it up into a free text sort of manuscript-like, abstract-sized manuscript. And then they submit it to the LitCoin server through journals, where a customized LLM would then extract the knowledge assertions it creates, and then it would create a knowledge graph of that assertion that were made from the text. And then what that does is that this would then allow the server to then give that knowledge assertion back to the author to make sure that it's appropriate and correct. They can double check it. So there's a built-in sort of review there. And then the publishers include the knowledge assertions in the peer review. So then if the paper is accepted, it will be published as a LitCoin in the chosen journal, and the curated knowledge then is included in a large sort of knowledge graph that is available for any researcher to access and to use. So really the overall goal is to build the generation of computationally accessible knowledge into publishing pipeline. And essentially what this allows is the researchers are already familiar with the journal process. All they're doing is writing an abstract. What then the knowledge graph is doing is it reduces the extensive human curation work that we do in doing literature reviews. That takes a lot of work. We do a lot of reading. Well, we can cut the time in doing that. So generating the ability to sort through that knowledge after the fact is highly valuable. So scientific knowledge, in a way, is really trapped in prose at the moment. It's trapped in these research articles. And the goal of LitCoin is to think about how it can become more computable, how can it become more connected, attributable, and cumulative at the moment of publication. So that's the idea behind LitCoin. Now, one of the ways in which we have used the idea behind how these knowledge graphs are so important for research is another program that we have developed called the Biomedical Data Translator. And what this does is it takes these knowledge graphs of these assertions that we found in the literature, and it's combining them with all sorts of other over 200 different data sources that are shown in this sort of spaghetti diagram. And all of these 200 different sources, we've created knowledge graphs to interact with them. And the knowledge graphs essentially organizes the connected data, allowing AI and machine learning to understand not just the isolated facts, but also the relationships between them. And it's sort of like how doctors relate to hospitals, or how a disease might relate to a particular therapeutic. And when you ask the AI a question, it's like asking it to explore a map of connected things, and it gives you a well-connected answer back. So that's the idea of knowledge graphs. And Translator has essentially a user interface designed with the biomedical researchers in mind. And at the moment, what it can do is it can support several types of queries, but one of the queries that I'm going to walk you through is what drugs might treat disease X? That's a simple question, but right now we don't really have a way to understand a lot of what drugs might treat disease X. If we had that answer, well, we might be out of a job. But here is a real-life example of a very rare disease called Shine syndrome. And Shine syndrome it's an acronym for a variety of different phenotypes that are presented with patients who have Shine syndrome. It's an ultra-rare disease. But essentially, it's caused by a mutation in the DLG4 gene that causes a loss of function. So there's a haploinsufficiency of this gene. So we asked Translator if they could think about, could we find treatments within all the vast literature that we have access to that might upregulate DLG4? That's a simple question that we asked it. So the Translator team was kind of hoping that they could find a key answer that came back. And it turns out that Translator, within a matter of about 30 seconds, returned many potential treatments. But based on the doctor's review, and this is a collaboration between a clinician and our informatics researchers team, our informatics team, the doctor's review of the output, he found that one of the medications that was on this list was called guanfacine. Guanfacine is a very common blood pressure medication, and it's a very safe blood pressure medication, but it's not known to treat this sort of disease. So the physician decided, well, this is a safe drug. Let's try this on this little girl with Shine syndrome. And they did. And so essentially, before treatment, you can see the highly detailed tests that we run, the coloring books of this little girl, and you can see the motor skills are not there. The ability to color in between lines is not there. However, after 5 months of guanfacine, this is what Peppa Pig looked like. And so the mother of this little girl sent a note back to us and said, "I've never seen Peppa Pig look so good." And this is the kind of idea where we need better ways of sorting through the literature that we have so that we can identify areas of opportunity like this. Now, this drug is not approved for for Shine syndrome, but now this might be the next step. This is something that we can work with the FDA on, talk to CMS about, and thinking about this as being the next approval for Shine syndrome. So we have many other examples that Translator has been has done in this kind of realm, and you're welcome to try it out yourself. There's a URL at the bottom of I'm still pointing to this. I don't know why. There's a URL shown at the bottom of the screen that you're welcome to take a look at and test out for yourself. It's a very unique kind of program, and we hope that this is one way where we can match existing drugs to new indications or new problems. So when we think also about literature and publishing, the idea of replication and reproducibility is often top of mind. And I wanted to just relay that NIH is elevating the importance of replication and reproducibility, and we have launched several programs now in support of this through a Common Fund program that we have. There's a prize competition. There are QR codes if you're interested in looking at these. And there's also a call for grant applications on the topic. And so there's a really big effort in thinking about how we can move the needle on replication and reproducibility. There is also an opportunity for community input on this topic. And I hope that everyone in this room can take a look at the request for information on that and think about how we can measure and reward scientific impact and replicable research. This is exactly what we want to hear from you on. So this group is primed to actually do that, so I hope you do. The request for input, I should say, is open until August 19th. You have time. You have a little over a month, so I hope that you take advantage of that. And of course, given the work that we do at NCATS and the need to always make sort of these go, no-go decisions on what we want to move through the translational pipeline, we take replication and reproducibility very seriously, and we always have. We've placed a high premium on this work. We hold workshops routinely. We have a program called the Assay Guidance Manual. It's not the most descriptive title for this program, but it really is a way of how we've pulled assays together to develop best practices for what those assays should be, to develop standards, and to help inform the community on what those should look like. And one of the programs is something that we have just relaunched. We've had this going for a long time, but during the pandemic, it shut down, but we're relaunching it now. It's called the Ring Testing Initiative. And given that rigor and reproducibility are instrumental in drug discovery because it bridges the gap between basic and preclinical research and translation into novel therapies, we want to understand how we can reduce the time it takes to really sometimes, we're chasing red herrings, and we want to reduce the time it takes to understanding how we're chasing what isn't reproducible and why. And so we're doing this next round of ring testing on basically a cell-based high-throughput screening, and we're seeding the cells, and then we're sending them to 9 different screening centers across 6 different countries, and we're asking them to do the same protocols on the same activities and understand what the outputs are for those. And that's one way that we can understand where variability comes from and how we can understand what are the factors that go into replication and reproducibility. And some of those can be very important for optimizing protocols until those results are reproducible. And then we would disseminate those findings to the research community through the Assay Guidance Manual. So it's been estimated that at least 50% of published work can't be reproduced in an industrial setting during discovery campaigns. And so this is kind of part of the problem that we want to understand or tackle. And so there's an urgent need for really setting these sorts of global standards for rigor in early drug discovery. But doing it globally is a way in which we can really maximize that variability in being able to do so. So this exercise is going to allow us to identify those sources of irreproducibility and solutions to address those sorts of issues. We kicked this off last month, and so it's probably, I expect these usually take about a year for us to get ultimately get results, so I expect that will come in about a year out. So stay tuned. So I've talked a little bit about publications. I've talked a little bit about replication and some of the ways NIH are addressing these issues. And I want to turn to how we work more broadly as an ecosystem that needs to maximize public funding and maximize private funding. Both are critical to our success. And in thinking about this, we also need to understand the problems. And so I highlighted a key problem for diseases. That is that we only have 5% of treatments or cures for all the diseases out there. So how do you work with over 10,000 diseases at the same time? This is the problem that we have. And the way that NCATS approaches this is that we think about this as essentially building platforms that can raise all boats for all diseases. So what are those platforms that need to be built? Well, we have a large focus on clinical trial innovation. I mentioned the different clinical networks that we have. We have a large focus on novel approach methodologies, those 3D tissues and organoid systems. We have a large focus on real-world data and AI and ML through some of the things that I've mentioned before. We have a focus on drug repurposing, gene delivery platforms, and rare diseases and pediatrics. And then we don't do as much in prevention and sort of this idea of disease reversal, but it's something that I've been thinking about quite a bit. But the way that we think about these platforms is that because these have the potential to raise all boats, it helps all of the other institutes and centers within the NIH, and it also helps our industry and private colleagues as well support some of their missions too. And as I've said, is if you solve one problem, well, that matters. But if you solve the bottlenecks that can help many more things, then that's going to change the field. So some of the priorities I have are I just mentioned. But in thinking about this prevention and reversal idea, it's sort of like turning prevention on its head. It's not, maybe we shouldn't be thinking about how do we prevent disease, but one of the key things that I've been thinking about a lot is because I have a partnership, our team has a partnership with NASA. And in this partnership, we have, over the last decade, we've developed tissue chips. We've supported the community in developing tissue chips on a variety of areas: lung, skin, muscle, et cetera. And what we've learned is that the astronauts that go up to the space station, they stay there for months at a time, then they come back to Earth. Well, while they're on the space station, they experience a lot of physiological problems. They have muscle problems, they have sleep problems, they have heart problems, they have kidney problems. So, but when they come back down to Earth, they actually revert. It takes time, but they revert. So there's something that's happening in the microgravity environment that hastens this sort of biological aging or pathology that's happening. And we see the same things in our tissue chip programs. So when we send our tissue chips up into the space station, they stay there for 30 days. In the first 3 days, we start to see pathology. We start to see cytokine development within those tissues, almost across the board. So that microgravity environment turns out to be really important for actually seeing this, because on Earth, this can take decades. So this is an important environment for us. But in thinking about moving from a disease state to a healthy state, that's a new paradigm in thinking about prevention. It's actually a reversal of the pathology. So what we know, I think now is that these tissue chips are perhaps a tool that we can start using to help us address this sort of problem. But for NAMs or 3D models or tissue chips, whatever you want to call them, for them to change translation, they need to become more elegant models. They need to become reliable measurement systems. They can't just be, we've built this model and we're going to use it for many different things, but we have to really understand the fit for purpose of what that model is going to do, and we have to understand the biology that we're asking the question about. Then we have the opportunity to understand the disease pathway and perhaps asking the right questions, which are different questions from understanding the disease pathway. How do we get back to a normal state? And could we start to really drive that? And thinking about what the previous panel talked about in terms of vagus nerve stimulation, I think this is an area where that's kind of happening already. What about other areas, and how do we support that kind of work and technology? So does that mean that there are pathological processes that can be reversed because we learned this from the astronauts, and that people can recover from a disease process? If we learn what's driving it, then I think that that's a reasonable question. So maybe it's the next big challenge that we have. Maybe it's too hard, I don't know, but but if we can prevent disease progression or even reverse that pathology, this is an ecosystem challenge. This is where it's going to we need to understand what parts of this problem need to be done by the private sector, what parts of this problem need to be done by the public sector. Where can we create the sort of the better measurements, the new biomarkers that are needed, the predictive AI, et cetera. This is where the entirety of the ecosystem is needed, and we all need to be at the table to think about this question, but I think it's worth thinking about. So I've talked a lot about preclinical stuff, and so I want to also talk about the clinical side in this conversation about innovation and redesign. So Seema set the stage very nicely for this, but clinical trials are where translation becomes real. It's also a spotlight where there are many challenges in getting treatments to people. This is where most of the costly failure happens in the work that we do. So this image, that image, these are where, this is from a JAMA article and that happened this year. And basically what it's showing is that the global early stage drug development is expanding. That's the bar graph. And so you see it going up. But the US dominance in this space is narrowing. The center of gravity is shifting more really towards a two-hub model, with important implications for things we care a lot about. We care about future FDA approvals. We care about investment. We care about workforce. We care about patient communities, and we care about patient access. That's why we need to care about this graph. Given NIH's recent emphasis on transparency and accountability and appropriate structuring of foreign components, the path forward in really trying to reset our curve so that it starts to go up, the path forward for this is around trusted collaborations. And this is, again, an ecosystem problem. So we want to have these trusted collaborations with partners who share equally high standards for the research integrity, for the data governance, and for the regulatory science. And those strategic partners can help maintain the global competitiveness. And that helps us also align our sort of respective expectations of clear disclosure, direct oversight, and responsible international engagement. So I think this is a key thing for us to keep in the back of our mind, and how we measure it ultimately means we need to be turning this curve. That's what we need to see. So acceleration is important in actually doing this. So where do we need to accelerate? Well, clinical trials, I think, are a key place for this, and because they are the bridge from discovery to care. Clinical trials help us determine what works for whom. They help us understand what's ineffective or what's harmful in terms of the approaches that we use, and they generate evidence that can inform care. So they do a lot of really good things. But again, most of the things that we put into clinical trials fail. So we need to do something about this failure rate. We're not getting enough answers from the clinical trials that we support. So we have these major challenges. We have too many trials that don't compete or don't complete recruitment, or are too small to be definitive. We have too many trials that take too long to get started because they're done sequentially. We have too many trials that are too fragmented, too generalized to the broader population, and we have too many trials that are disconnected from decisions that patients, clinicians, healthcare networks really need to be making, or at least adopting into their workflows. So to get back to these sort of issues and how to address them, there was this phrase that was used during COVID, and it was the small, crappy trials, that we were doing small, crappy trials. We were all guilty of this, by the way. It was public, private sectors were all guilty of doing small, crappy trials. And it became this memorable soundbite at the time because it captured a real frustration. And that real frustration is that really small is not the problem. There are lots of small, very effective clinical trials, and they're very good. So small is not the problem. But the crappy part, we need to be thinking about that a little bit more. Not all of them are crappy, but design how you do these clinical trials. What are the decisions you need to make? That focus, there's a lack of focus sometimes in that, and the incentives are not necessarily aligned in how we do clinical trials. So there are problems, and those problems are centered around the notion that Too many trials are not informing those decisions that we need to make. And so I've listed some of the common weaknesses in trials here and what to do about those common weaknesses. But I'm going to pick on one, and that is that they're not harmonized with related studies. That's the one I'm going to pick on. There was a study done on uterine fibroids a couple of years ago, and there are 2 surgical approaches for uterine fibroids. So the trial was to compare those 2 surgical approaches. It was a small trial, and it was a negative trial. Didn't see a difference between the 2 approaches. So the PI published that paper, that negative trial, and I'm really glad she did. She was part of the CTSA program. I'm really glad she did. After it was published, she got several calls from other clinicians across the country who said that they did the same trial, they came up with the same answer, but they decided not to publish it because it was too small and inconsequential. So instead of them publishing it, the field lost time, patients were enrolled in duplicated trials, and the evidence remained weaker than it really needed to be. So now those groups are actually getting together to do a meta-analysis on the findings for a much more robust understanding of that determination. But the point is that this highlights a key problem in a nutshell. The point is that not that the trial was small and not that the trial was negative. It was just not connected, visible, or cumulative to other questions that people were asking in the same ecosystem. And that's something that we need to surface and elevate. So the challenge for us isn't just to think outside of our box. It is also to build a system where evidence doesn't stay trapped inside of our own little bubbles that we're working in. So I think a lot about how we separate small but important from small and uninformative. That's the distinction that I think we need to be asking ourselves. And then we build the infrastructure that makes every trial more decision ready. And when I think about this as sort of re-engineering the enterprise, I can't help but reflect on the fact that we have an opportunity. If we're re-engineering, we have an opportunity to think about accelerating and that urgency that's on the table. It's not necessarily about going sort of faster in a way in one aspect of that pipeline, but it's sort of about going faster in the areas in between where that pipeline exists. And so the planning, the readiness, the design, those all are things that we can do better at. There have been incredible advances in new modality, precision medicine therapeutics. I'm actually going to show a little bit on that. And it's telling us that the science, again, is moving way faster than we can keep up with. So we need to redesign the path from promising clinical evidence by removing the delays that aren't adding any value. And if we don't do this, then we're going to lose more than time. We're going to lose momentum. We're going to lose talent. We're going to lose patient trust. We're going to lose industry confidence, and we're going to lose the leadership edge. And that downward trend that we have right now and that in clinical trial and preclinical development is certainly suggesting that. So that's why we need to have this conversation. And so in thinking about this, in the clinical trial landscape, actually in the translational science landscape, essentially we do a sequential approach. We think about it in a sequential order. And these are exactly the kinds of problems that NCATS is trying to address, not at a one disease kind of a way, not at a one protocol at a time kind of a way, but the common bottlenecks that slow the translation across these diseases, interventions, and communities. And so right now we're sort of stuck in this top version. We're stuck in the sequential pipeline. And if a spot in that sequence breaks down, then the subsequent one doesn't happen, much like a resistor. And the lesson that we have from the chip technology community is not that everything needs to move through one wire faster. That's not the idea. The idea is that speed comes from the architecture and how it's built. So we need to think about how we rebuild that. And so the parallel approach is we don't have to parallelize everything, but where we can, we should. And so things like when we're building the IND application and the protocol development, we need to be working with our patient communities and getting them into the trials at that point in time. We need to be writing our IRBs as well. So these are things that we can start to parallelize and advance them for moving faster. So to be clear, parallelization is not about shortcutting the system. It's it's about sequencing what absolutely must be sequenced, but picking up the pace by doing things in parallel where we have the opportunity to do that. So it's not less rigor, it is less waiting. And I think that's a key area that we can go into. Now at NCATS, we've been doing a variety of these activities through the CTSA program and the rare disease networks that we have, real world data programs that we have. I'm not going to talk about this slide a lot, but just to show you that we have infrastructure innovations through initiative development, and the impacts that they've had is that ultimately we've been able to do faster clinical trials. One of the key areas in this is something called Smart IRB. We have over 1,500 signatories on our Smart IRB, and that helps different multi-sites rely on each other to do clinical trials. And that has reduced the time for IRB approval from the matter of 9 months to a year to the shortest one, which is arguably too short and not really right, 18 minutes, but averaging now at about 1 to 2 weeks. So this is the idea where we can speed things up and move things faster. And so these are the kinds of things that we're trying to do. Now, somebody mentioned before that there was a program in DC that was launched called Operation Trailblazer. Operation Trailblazer. I was involved in this program, and I'm still involved in this program. This is exactly what this is meant to do. Is around the table is FDA, NIH, CMS, and other HHS agencies, as well as academia, pharma, biotech, venture capital, and nonprofits and patient organizations. That's who's around this table. So this is an ecosystem problem that we're trying to understand how we can row in the same direction together. So the idea here is that public investment accelerates private innovation, and the public sector can think about things that are hard and challenging and take a long time: measurements, standards, natural history studies, cohorts, et cetera, so that the private sector can build on that, can leverage it, do the therapeutic discovery and optimization, do the product development and launch those. That's the flywheel, but it takes all of us in that flywheel to make it happen. And it's also bidirectional. So conversations like this that we're having, where everybody's in the room, these are the kinds of conversations that help us. So these are just some examples of showing how the ecosystem is working. I'm not going to dwell a lot about these, but essentially no individual health system would create a national sort of real-world data network, but that's something that we're working on. This is built from the CTSA program as a whole. They came together, and this public investment now is creating an ecosystem that academia and industry can now build upon. The other example of this is the Rare Disease Clinical Research Network, where we're bringing together a variety of different communities across the country to study rare diseases. And through this network, they've developed 13 new drug approvals to date for 12 different rare diseases. That's pretty impressive for a 20-year history of this program, given the length of time it takes to do this. And that's because we have a large number of people at the table, including patients, to help us do this. But we're sharing the costs of that infrastructure across the network to help raise all boats. Gene-based therapies for rare diseases. This is a huge area of activity, from gene editing to gene therapies to platforms that are used to build these kinds of programs. The NIH has this program called the Somatic Cell Gene Editing Program that is laying the groundwork to rapidly develop treatments for other rare genetic diseases. And one of the ones that I'll talk about today is baby KJ. And you probably have heard the story, at least I hope you have. If you haven't, you may be living in a cave. We should talk about that. But baby KJ has this disease called CPS1 deficiency that was corrected with gene editing therapy. So because we have built this somatic cell gene editing program, everything was there. The infrastructure was there. This baby came into the University of Pennsylvania. The physicians were a part of the somatic cell gene editing program, and that was the thing that happened. So it took the family, the patient, the clinicians, and the funders to really come together, and the FDA to come together to make this happen. And essentially what they did, he had a very severe form of this disease, and within a few months, they needed to sequence this baby to understand what was wrong. This is a urea cycle disorder. We've known about the urea cycle pathway for a century, yet we still can't treat it well. We have 4 therapies on the market. All of them scavenge nitrogen. None of them actually get to the heart of what the disease is until now through gene editing. And so now, through this program, we created the gene editing mechanism to treat the specific mutation within baby KJ, deliver it to him, to his liver, in a matter of 7 months, working with NIH and the FDA to make that happen. And now he's running around, literally, and doing great. And his outlook was not good before that. So this took the whole ecosystem to come together and make that happen. But the idea, too, here is this is kind of a one-off at the moment. We need it not to be a one-off. So how do we create gene editing platforms for everybody? And the idea is now we're working with the FDA to understand, well, gee, if the urea cycle gene list is, there are over a dozen genes in the urea cycle disorder. So if we know that, then maybe we could use a master protocol to think about one toxicology biodistribution study, one animal model, or even one novel approach methodology that we can use for the evidence collection for all of these different diseases of the urea cycle disorder. That's the impact that we're looking for, new ways of thinking of how to move these projects along. So this is, I think, an exciting area. I talked a little bit about drug repurposing earlier. I talked about this on the side of sort of existing therapeutics to treat new indications. Well, the same thing is what we're thinking about for gene editing and gene therapy, because it's really about the delivery repurposing. The way we deliver the genes or the gene products or the machinery to edit the genes, that's the cargo. But to deliver it to the right cells at the right time, at the right place, that's how we need to figure out how we can repurpose that delivery. Sort of like the post office. You buy a box at the post office. If it fits, it ships. So kind of that same idea. We want to reuse that sort of infrastructure. So this is my last slide. Sort of on thinking about translational science, innovation, and redesign. How do we accelerate this so that we all succeed? This is just a list that I, off the top of my head, that I came up with in thinking about this talk. But the ecosystem is not about who owns which piece of this. The future of translational science will be about understanding which problems need to be shared, which bottlenecks are public goods that need to be broken, and which are the private bets that need to be encouraged, and how we connect them to the right capital to get to the right evidence, while also being accountable to the questions that we're asking, without losing time, without losing rigor, without losing that trust in the patients who need us. The ecosystem that connects the right pieces, that funds them appropriately and makes what learns reusable, will be what makes it sustainable and robust. And so of this long list on this slide, I want to highlight 3 that I think of as I'm thinking about at NCATS. And one of them is to fund infrastructure that compounds, that is a force multiplier. The second is to publish knowledge that accumulates knowledge so that we can start making those connections so that we're learning without having to do a deep literature search for a question that we're trying to ask. And the third is to design translations so that rigor and speed reinforce one another, and so that the public and private sectors can really navigate the areas in a way that we've never done before. So thank you very much for your attention. I'm happy to answer any questions if we have time.
Sorry, I was hidden behind the pillar, but great presentation. I was very interested in the translator in this approach, because, I mean, this is our dream probably, to take a drug that is approved and then just test it on something where we don't have a treatment. So, but one thing I was wondering about, are there thoughts about having some sort of a closed loop system? So basically, let's say I'm testing this drug on a patient and it helps. I can put in this information into the translator, or maybe most of the times it does not work. So I put this information also into, I mean, you mentioned this as well. There's a lot of data that's negative. It's not out there. It's not published. Is there a way or plans to implement this into the translator so it gets better and better with time? And then maybe, this repurposing can be even better one day.
That's a fantastic idea. The short answer is yes. That's something that we can do. We haven't done it yet, but it's something that we can do. What I will say is there's another program that we have called CureID. It's a partnership with the FDA and NCATS. And CureID is essentially a crowdsourced platform where you can go in with a new drug that was not intended to be used for a particular treatment. We know that this happens a lot, and so we wanted a way to try to capture that in a crowdsourced way. And so we're using that as perhaps one of these ways in which we can get that all of those sorts of data. What are those outcomes? Was it negative? Was it positive? And maybe perhaps use that to help feed into something that translator could ultimately use. So connecting those two programs might be the key that we need to get at. So thanks for giving me the idea.
Thank you so much, not only for the talk, but for all the work that you and NCATS does. So if you could tolerate a question that you didn't address. So in my experience in translational medicine in big pharma, one of the major bottlenecks is the lack of PD biomarkers.
Yeah.
So, what academia can give us not only is the discovery of the pathogenesis and the mechanism of action, but how do we know how do we predict that the drug is going to be effective? And can you tell me what NCATS is doing in that regard?
Yeah, it's a great question. Biomarkers are so tricky. And this is where I think NAMs are going to be really valuable. And it's why I say that we really need to make them fit for purpose and understand the biology that we're looking at. How we build the NAMs is going to be important to be able to do that. I think NAMs can help us get to the granular nature that we need to get at for what those biomarkers do. I think part of it is technologic. We're not able to measure the right things, or if we know what we need to measure, that's one thing. But if we don't know what we need to measure, we may not be able to detect that level of sensitivity to understand that's the thing we need to measure. So one of the things that NCATS is doing is actually delving into quantum sensing in a way that we can use quantum sensing technologies to put into novel approach methodologies or new model systems, and think about what are the areas of detection that we should try to be amplifying, and in an effort to identify new ways of getting at biomarkers. But I think they're very noisy when we think about outcomes, and the FDA is very particular about what they want to see in biomarkers. So it's always that's a key bottleneck. And so a focus on this is really thinking about how can we increase the sensitivity of what we're trying to look for. So that's one area how NCATS is trying to approach it.
A fantastic talk, Joni. I just want to emphatically agree that infrastructure challenges can present a huge bottleneck. A few years ago, as part of the work that I was doing leading R&D for Disney, I had worked on the COVID defense with vocal diagnostics. And without getting into the details, that really didn't get where it needed to be simply because of data harmonization issues. People were collecting different data for no reason. The IT systems were doing things like using different anonymization hashes so they couldn't talk. So I think there's a lot that could be done here. But the question I have for you is related to this. It seems to me that diagnostic technologies are already at the point where huge values can be happening in society, but they're not. I'll give you one example. I believe 57% of Americans with Alzheimer's are currently undiagnosed or unaware of their diagnosis because 97% of primary care physicians just don't proactively screen for it. But you can detect this with a 30-second vocal screen. So some of the initiatives I'm involved in related to ARPA-H Prosper are to leverage these non-invasive diagnostics, and I think we can radically transform the entire system. So my question to you is your program's a good potential home or collaborator for these initiatives that are kind of underway? And if so, I'm very happy to talk to you.
That's a great point. I didn't talk a lot about diagnostics, but they're there. Absolutely. And I couldn't agree more. Thinking about AI in just the doctor's office, it could transform how we start to diagnose. There are still ethical concerns around all of that. they're overcomable. But I think that this is an area that is ripe for really advancing, and so I'm excited that you're talking about it. It's not just Alzheimer's; it's Parkinson's, it's rare disease, it's a variety of things, and so you can use even your watch to detect epilepsy episodes. There are a variety of these technologies that are out there, and I think those are going to be very helpful. So how we use wearables, how we use AI, these are great questions. For me too, in thinking about rare diseases, on average, it takes about 7 years for a rare disease to be diagnosed. And you talked about sort of Alzheimer's disease and other common diseases that take a long time to be diagnosed as well, because usually you don't go to the doctor or whatever it is. But this is time. This is time where those diseases could be malleable. So if we can reduce the window of time of that using diagnostics better, I think that's also going to help us think about this idea of prevention to a more severe state of disease. So these are great ideas. I'd love to talk more about them.
Thank you.
Thank you, everyone. A round of applause for Dr. Joni Rutter, please. Thank you. Thank you so much. That's amazing.
Thank you.